In this course, we will talk about functional characterization of a list of genes. So we will try to find the answer on the question, what is the biology behind this list of genes? So more and more omics experiments are generating lists of genes. For instance, a list of differentially expressed genes that was identified in a bulk RNA-seq experiment, or a list of genes that all have a mutation in a tumor obtained by exome sequencing, or a list of proteins that are thought to interact with a bait obtained by doing a proteomics experiment. So all these omics experiments are generating lists of typically thousands of genes that are very hard to interpret because they're simply too big to look at every gene individually. And that's the most heard question after people do bulk RNA-seq, for instance. Yeah, I have now my differentially expressed genes, but what's next? Which one do I have to pick for further research? So it's very hard. So one of the things that can help you is to functionally characterize the list of genes. So there are two approaches here that you can follow. You can see what the genes in the list have functionally in common. Do they all play a role in the same biological process, for instance? But on the other hand, instead of looking at function, you can also look at regulation. Or they're all regulated by the same transcription factor? Do the sequences of the genes have motifs in common? So in this course, we will talk about these two aspects, but we will start with the functional characterization. So we will see what the genes in the list have functionally in common. So for instance, if 60% of the genes in the list are all involved in the MAP kinase pathway, then you can assume that this pathway is playing an important role in the treatment that you did in your experiment. So by going from genes to pathways, you make biological interpretation much simpler. You just look which pathways are occurring a lot in my list. These are the pathways that are playing a role in the conditions that I have studied in my experiment. There are different methods for doing functional characterization of a list of genes. And basically you have to make a distinction between methods that are based on enrichment and methods that are based on topology. So what's the difference between the two? In the enrichment-based methods, you see pathways as simple bags of genes. Each pathway is a list of genes, and the order of the genes in the pathway is not important. In the topology-based methods, you're going to take into account the structure of the pathway, the topology of the pathway. So there, a pathway is a list of genes or a list of proteins, but the proteins have a certain order, a certain location in that pathway, and you take that into account. Now, we will start with the enrichment-based methods because these are the methods that are mostly used for doing functional characterization. And here again, you can make a distinction between two types of analysis. On one hand, you have the overrepresentation analysis. On the other hand, you have the gene set enrichment analysis. So we will start with the first,